I'm going to make the point that we are now at the cusp of exponential growth of bad medicine. And part of what we've been celebrating today is contributing to that exponential growth. The good news is some of the same tools are going to allow us to overcome that exponential curve. So here's a spoiler. Here's what I'm going to tell you over the next 25 minutes. The healthcare system does not know how to pay attention to you. Fundamentally, it's been overwhelmed by data. Doctors have been overwhelmed by data. More data is going to overwhelm it more. And the process automation that is currently available just makes it worse. The healthcare system cannot be accountable if it cannot count. And right now, it cannot count even very basic things. The healthcare system casually and causally hurts us both by inattention, false conclusions, and false wisdom, some of which we've heard today. Biomedical research often misses the big picture. A deep and broad information commons, which I'll describe, is a key to the solution. We need more quants. And just to keep you awake for these 25 minutes, I'm going to ask you a question to see how many quants are there in this crowd. Do you need to go back to school? Multidisciplinary teaming and an imminent, truly disruptive process and automation will, in fact, overcome the exponential badness. So a Malaysian airliner disappeared, and the whole world was activated to figure out where it had gone and what had happened. At two of our top hospitals in Boston, the following peak of heart attacks happened. So this was over the baseline, an 18% increase in heart attacks. And so what we were flying into this mountain of morbidity was not a plane load, but 1,000 plane loads, 10,000 plane loads of patients with heart attacks. And guess what? No one ever noticed. It's only when we did the analyses through the electronic health record of the data that we became aware of it. So it's like 1,000 Malaysian 747s disappeared without a trace. But in this case, we caused it. We actually shot them down. We gave Vioxx, and we are the cause of that mortality and morbidity. We caused it. You'd be giving me a standing ovation, maybe, if I told you that we had caused a 5% decrease in heart attacks. We caused an 18% increase in heart attacks, and we didn't even know it. And it was knowable. Why do we know this? Because Kaiser Permanente took it off their formulary early on when they saw early papers suggesting that there was cardiotoxicity. That's our healthcare system. And guess what? I have many other papers which I can cite, some of which we've done, which tell you this is happening now, today. We're not paying attention. You know, all these measures that we're hearing about, readmission, this and that, there are huge signals that we're missing if we could only bother to be creative and look. Here's one patient coming into the healthcare systems. Ben Rice, one of our faculty, aggregated all the ICD-9 codes, discharge codes, across all of Massachusetts. And so you see this one patient coming into the healthcare system at the center of the circle. Coming towards the outside of the circle is the present. What's the diagnosis of this patient? Domestic abuse. What was Ben able to show in 2009? That we were able to predict very accurately, on average 2 years before the healthcare system was aware of it, that there was domestic abuse going on. And not with the odds ratios of 1.3 or 1.5 that we love in genetics. Huge odds ratios and highly effective. What kind of healthcare system lets domestic abuse, which actually is very plain to see once you bother looking at the record, but who's going to have the time in that 30-minute patient encounter? What kind of healthcare system is it? We are not paying attention. I'd love to think that doctors are synthetic and can deal with uncertain and chaotic information, but we have no evidence that on average they do. I coined a term back in 2006 called the incidentalome, the biggest ohm of them all, bigger than the genome, the proteome, even the microbiome, Larry. And what I've said was very simply this: even if we have a test that was 100% sensitive and 99.99% specific, if you're going to measure 10,000 things at the same time, 60% of the United States population will be falsely positive, and this would make the false positive disasters of mammography and prostate-specific antigen looked like chump change because everybody would end up being surgerized for no particular reason. So I predicted this back in 2006, and sure enough, you've all heard about sudden death on the basketball court, and we're all worried about it for this disease that's 1 in 500, hypertrophic cardiomyopathy. So here's big data coming into the scene. National Heart Lung Blood Institute puts out a bunch of exomes, around 7,000 exomes, looking at the frequency of all these alleles. Well, guess what happens? You look at those putative causal variants, and what we see is some of them are very common. How common? 25% for a 1 in 500 disease. And which population are they the most common in? African Americans. And who are the ones who are on the basketball court? Mostly African Americans. And what are we doing? We're implanting defibrillators into them for these common variants. Why are we seeing this high rate? Because all the studies were done initially on white people. Because we tell in genetics, you need a case control population matched. All well and good, but now we've done something not good. And that's just one gene. I heard we have a lot more of them. So I've been involved in the study of autism. And if you talk to neurobiologists, they show you this nice complex diagram of signaling at the synapse. So synapse, the space between neurons, that's where all the action is happening. And they found all sorts of wonderful mechanisms. But when I did a gene expression study looking at what genes were turned on in the blood, I was pleased to see neurotrophin signaling was different and long-term potentiation was different. These are known processes in the brain having to do with memory, for example. However, I saw a bunch of inflammatory signals, and I was thinking to myself, what's with that? So then we looked in the literature, and it turns out there's a whole parallel literature that you'd never hear from the neurobiologists about inflammation in autism. And not only that, if you looked at all of Denmark, mothers with rheumatoid arthritis and fathers with type 1 diabetes, autoimmune diseases, have increased risk of autism. Mothers with a high gestational CRP and a measure inflammation, higher incidence of autism. Intra and peripartum infection, higher incidence of autism. Nothing to do with a primary defect in the synapse. And a mouse model of inflammation developed by the late Paul Patterson from Caltech looks just like any genetic model of autism, except all he does is cause sterile inflammation in the mothers when they're pregnant, and the kids look completely autistic. I'm not saying that there's not another mechanism that looks like classical synaptic pathology, but there's a whole immunological mechanisms that you don't see. And in a recent paper, I just did a very simple set of Venn diagrams. Shown in N is all the open access literature, that's NIH-funded literature, involving autism, synapse, and genetic causes. That's N. I is the same thing, but it's now immunological causes. Look how not overlapping they are. Furthermore, look at the literature they cite, I cite and N cite. Almost completely non-overlapping. These are two major, heavily funded research establishments that essentially do not recognize each other. And they're talking about the same disease. This is not unique to autism. So, part of what I've been doing over the last 10 years is figuring out how to use healthcare systems as living laboratories. So we've created something called I2B2, which is an open source system that allows distributed queries across multiple healthcare systems, and it's been adopted now by over 100 academic health centers internationally, 80 of which are in this country. And so with some of my friends, I said, let me do a distributed query across approximately 6 million individuals for a subset of those hospitals and see the kids who have autism and what can I discover about them. And so these were only about half percent of the hospital population, which is probably a low estimate given the fact that we think it's about 1% prevalence. Male to female, 5 to 1. Typically it's thought to be 4 to 1. And involved, importantly, 5,000 other medical diagnoses. We talked to a developmental medicine specialist. They think of autism as mostly having to do with these behavioral issues. But we found 5,000 other diagnoses. And work that was done by Finale Doshi, we just crammed together all the patients and lined them up so that there was a bit switched on if they had one of those 5,000 comorbidities and a bit switched off if they didn't. So they have these bit vectors for each of the 6 months. And then we clustered them together. And in plain English, we asked ourselves, which patients resemble another? Are there natural clusterings, natural conglomerations of these patients. And to make a long story short, across this network that we had assembled, we found one group of patients who had a lot of epilepsy, up to 80% epilepsy. Another group of patients who had a lot of infections and inflammatory bowel disease. And another, so it looks much more like an immunological disease. And yet another group with autism who had a lot of psychiatric disease, ADHD, schizophrenia, and so on. These look like 3 separate diseases. And yet our genetic studies treat them out as if they were one disease. And just by looking at these large data sets where we've overcome the technical and sociological problems of sharing data across large health systems, all of a sudden we see granularity in the diseases that are going to change the way we diagnose and treat these individuals. But clinical phenotyping is not only phenotyping. We've taken together hundreds of thousands of microarray experiments. So these are experiments which measure which genes are turned on and off in multiple tissues. And because of the way NIH works, thankfully, hundreds of thousands of these experiments have been deposited. And so we just parsed all the descriptions of the experiment, took all the data from these freely available experiments, and were able to actually create a landscape of differentiation, disease, and cell type across all these experiments. And as a result, for example, for something like autism, we're able to take iPS cells, that is, cells taken from your skin, de-differentiated and turned into neurons. So it's basically a neuron that looks a lot like you in the dish, because we took it from your skin. And then we can actually project it by seeing which genes are turned on and off into the greater crowdsourced knowledge of what is a healthful and non-healthful, differentiated, not differentiated cell, neuron specifically, and see, where you fall in the spectrum of disease and how different small molecules might affect your disease. So what we're converging towards is a vision that a National Academy report, which coined, I think, the term precision medicine, or if it didn't coin it, popularized it. I had the privilege of being part of this committee. And what we showed was that, what we hoped for, rather, was that just like Google has turned enormous amounts of value out of the ability to mash together multiple modalities around geography, all with the same coordinate system, longitude and latitude. What we need in medicine is an information commons where we're similarly bringing together different data types, but using the patient as the common coordinate system. And as you've heard today, that is not an easy or always sociological doable goal. And furthermore, as we pointed out in a paper in JAMA that we published this May, it's not just healthcare system data. The conventional healthcare system is incredibly important, but all these other data sources, such as the blogosphere, where there's a lot of unstructured but important information, and all those other sources of unconventional data, all those data sources are not indexed by the same patient record. Even in a fully socialized medicine country like Denmark, you can't link your patient record to your tweets, for example. But we're going to do that with appropriate patient consent if we are going to see the patient more holistically. What about public health data? We just heard about Ebola. And so for public health, this has been the typical cascade of interactions. And really the goal has to be, if we're going to be nimble, to have this kind of interaction. And so my colleague, John Brownstein, in Boston, has been quite effective, and I'll just show you a few of the implementations he's done of taking all these varied data and actually making something of them. For example, using them to pick up disease earlier and cheaper than traditional public health monitoring. So I grew up in Switzerland, where the headquarters of the World Health Organization. I always assumed there was going to be a big DEFCON 5 room with all the screens showing all the outbreaks being tracked. Well, guess what? It doesn't exist. But John actually has created it, and it's actually now being used by the CDC and by the WHO. And what it does is, it's called Health Map. And Health Map works every day, every hour, and uses a multiplicity of sources in 15 languages. And it takes all of these and generates alerts over 10,000 locations and identifies both the organism, the location, the extent of it, where, and then across multiple species, and then categorize it according to the validity, according to its timeliness, and then actually, in the end, turns this into breaking and warnings that are currently available every day. And as I said, the public health institutions use this now routinely. And it's a nice paper showing that he was able to track H1N1 nicely from the index case using these means. But even for things like that are less potentially dangerous, like food outbreaks, the Centers for Disease Control and Prevention approximately report 1,000 a year. He reports through his system 10,000 such. Using what? Using things like Yelp. Now that might sound crummy, but what's interesting is when you look at the distribution of causes through these unconventional data sources, they look just as the same as the CDC distributions. Okay, here's where I told you had to wake up. We asked a question of our Harvard medical students, residents, and attendings that had been asked 30 years before. Let's take a disease that our leader, Dan here, is suspected of having, zombie disease. And we have a test for zombie disease, and it's actually pretty good. And it's complete sensitive, but has a 5% false positive rate. Zombies right now are not yet that prevalent, so it's only 1 in 1,000. So if Dan tests positive for zombie disease, how likely is he to have zombie disease if he tests positive? Now, here we go. Hands up. Does he have a 100% chance of having zombie disease? Does he have a 90% chance of having zombie disease? Does he have a 70% chance of having zombie disease? Some hands going up. Does he have a 50% chance of having zombie disease? Some hands going up. Does he have a 40% chance of having zombie disease? 30%? 20%? 10%? 5%? 2%? All right. I would say roughly your peak was around 15 to 20. And when we asked this of the Harvard people, this is what we found. The most common answer was 95%. The correct answer is 2%. Let me work it through for you. 1,000 individuals, 1 true zombie, 5% false positives, 50 false positives. So for every true positive, we have 51 positives. So it's 1 in 51 chances of getting it right. So this is not genetics. This is zombiness. What if we have to do this across thousands of genetic tests? Are you not going to be a little bit worried that the doctor doesn't get it right? Because guess what? Doctors are no better than us at being Bayesian update machines. And by the way, we published this in JAMA Internal Medicine, but 30 years before, it was published in New England Journal of Medicine. And the Harvard doctors, if you want to see the cup half full, were no worse 30 years ago. But what kind of system is it where we can't actually do basic statistics? I tell you what kind of system it is. It's where we're human beings, and what we're going to do is have to rely on process automation. So how is 1980s technology, which is what our electronic health records are, going to overcome exponential badness for 21st century medicine, where if we have to be these Bayesian update machines across millions of variables? How are we going to get there? I was amused to see on this website this picture that was described as doctors and residents working with patients at the JFK Clinic in St. Louis. Well, guess what? That's real. That's present now in most hospitals. You see a bunch of people who previously might have been talking to patients, and they're up there like monkeys, typing away to get the documentation right so that they can get paid. That's the doctor-patient encounter without the patient. That's what process automation today has created. That's what we've just invested $44 billion into happening at the national level. And with my colleague, Ken Mandel, We actually tried to warn back in 2009 that this was going to happen. We said, look, this is not the right technology. It's just going to make us data drones and not data users in decisions support. But we actually tried, and not only did we publish this paper, we actually tried to do something about it. With funding from the Office of National Coordinator, we created something called a SMART platform, which I'll spare you the technical details. Think of it as a thin layer of HTML5 on top of a common API on top of existing electronic health records. And early on, we showed that, for instance, we could take a beautiful user interface that fully compiled in PDF as part of a contest from Wired magazine, and we actually turned it into living, breathing code within 1 week and made it available across a multiplicity of systems, PHRs, personal health records, electronic health record systems, and data repositories. But I thought we were going to go down because I thought the EHR vendors were going to resist and not adopt this, and I was wrong. At the largest conference that happens in the year, the HIMSS conference, several vendors, without us knowing that was going to happen, have taken the SMART API and are implementing, so the same apps are running on multiple systems. But here's the real significance of it. What this allows is you, the larger developmental and innovating public, to actually innovate and create apps like perhaps the domestic abuse app to actually do something different. Because right now, however good or bad the EHR vendors are, they're a limited resource, even if they have 10,000 developers. If you allow the whole world to innovate, just as the iPhone or Android have allowed us to do, then we're going to see that transformative effect happen. So I honestly believe that through something like this, the ability of apps to run against our data, and both the big data and our individual data, is going to be transformative, because merely allowing a lot of smart people to access process automation medicine will be transformative. Wrapping up, here's more source for joy and hope. We ran a competition a few years ago where I was concerned about clinical grade genomic sequencing. And I was worried that it was not happening at clinical grade and only top groups would be able to perform it. I arranged a conference at Harvard, and immediately the conversation devolved into yet another standards discussion. Disgusting. So instead I said, let's have a contest. And we'll actually create teams on the fly that's going to do this. And so with a small prize of $25,000, we put out the data of 3 trios, 3 families, each of which had a very sick child. And we had the genomics and of the parents and the kid and the full record available to teams. 30 teams competed internationally. And what was amazing is within 2 months, they had actually found the cause of disease in 2 of the families, including this child who had gone without a diagnosis for 11 years. And here's the rub. 2 of the winning teams made the diagnosis, and these were hospitals where the patient had previously been. What's the difference? They had a multidisciplinary team of quant nerds, doctors, geneticists, lab people working together. And it turned out a lot of teams used the same pipeline, but having both an interesting amount of horizontal thinking on the geneticist's part and a lot of good vertical thinking on the bioinformatician's part, we were able to actually get to a solid diagnosis that could not have been done otherwise. Now, this is something that we can turn into practice, but we need a different kind of medicine. This is the birthday cake that this kid gets after 11 years. Can you imagine? Here's another one. I was in the emergency room with my mother. She had woken up and she called me. She's a tough old bird, and she was complaining of vertigo. She had trouble standing up, and she had slurred speech. And so I drove her over to her apartment and brought her to the Brigham Women emergency room. And I'm known now. I'm old enough, unfortunately, to be well known at Harvard. And one of the emergency room doctors comes, and they take very nice care of us. And just to show me, like, he gets what's going on, he tells me which cranial nerves he thinks are involved, and these are going to be confirmed shortly by the MRI. I said, okay. But I'm trained as a pediatrician, And so, but it didn't seem right. It didn't seem like the right diagnosis, not what I remembered from medical school. So I used this really great artificial intelligence program, and I hope after this lecture, you too will use this great artificial intelligence program. It's called, let me see, Google, yes. And I typed in stroke, diplopia, dysarthria. So to give it some credit, what I did provide was 3 techno words. I was told that in the first year of medical school, they shoved 20,000 new words into our heads. And it made the right diagnosis on the first shot that the MRI confirmed. By the way, she bounced back immediately. She's the kind of person who says, I beat Hitler and I beat Stalin, which she did, and she beat this too. But the point is, the knowledge is out there, and we're just not doing yet a good enough job putting it to practice. And most of our young doctors, when they're figuring out what movie to go to, they have a Google reflex. When they're in the clinic, they don't. Why is that? So one important thing that you can do, one very important thing you can do. Many of you may have seen the article about this guy, Peter Dreyer, who got a surprise bill of $117,000 because some neurosurgeon whizzed by him and made an additional consult, and they charged another $117,000. The follow-up story from the New York Times is that he asked for his medical record back. And this is not 10 years ago. This was this past month. And it took 6 weeks and lots of chest beating to get that medical record back. So here's what you can do. There was a big investment, I just told you, $44 billion on the current administration for electronic health records. And one of the promises was this, something called the blue button. It was going to give you all your data in a formatted XML format. So here's what you want. You want to go to your healthcare system and ask for the blue button download of your data. Because under meaningful use, this thing is supposed to be happening imminently. In some places it's already happened, it's supposed to be happening imminently. And you could say, I want my data through the blue button. And ask them why it's not available. If you can do that, all the stories we've heard about today are going to be super accelerated. Because this will give us all our data. Make blue button happen. There is a commitment in writing by these vendors to actually be compliant with this. Make it happen. Ask for your data through the blue button. Thank you very much.